Hey yo, in today's video we're going to cover some basic usage of the Form Bio platform. The first thing we'll talk about is data, adding data to the platform, managing your data, and making your data usable for the different analysis and workflows that you can do on the platform. So first, we're here on our home page of any project, and you'll see that there's a quick data tab to add data to your said project. If we click that, there's three different ways to add data. The first one is a simple upload from your own computer. This allows you to select files or folders and then import those into the platform. You can also use one of our tools to import data as well, whether that be custom data that can be imported through our Form Bench app called Form Shell, or if that be using our Form Bench transfer data platform tool.
Hey Jeremy, can you tell me why I would want to use the form data transfer tool or the custom data versus local upload? So for example, are there size of my data that would be advantageous to use the data transfer tool?
Of course, yeah. A lot of times scientists have huge data sets that they need to transfer from a sequencer or anything like that. In that case, it would be better to use one of our transfer tools rather than having to download those files to your local computer and then upload them to Form. With our tools, it's a quick and easy way to transfer them directly to the platform and get straight to the analysis, cutting out the middleman. So now let's go take a look at the data as a whole. Once you've imported the data, it lives here in this data tab. This functions a lot like your typical file management systems. You can have multiple different folders, each with folders within them, and you can also store your different data files here. This is a quick and easy way to manage data and make it usable for the different workflows and tools. As you can see within workflows, within our data project, we have the ability to make new folders given any name, as well as add data directly here by selecting files or folders from our local computer. So that kind of is a quick and easy file management system. So in order to run a workflow, I'm on our home page here, and there is the next, little quick tab that we've got, Run Workflow. Here it will take us straight to our workflows launch page, which is a list of all of the different workflows that Form has to offer. Some of them are in-house curated, and some are more open source that are brought in from the community. Along with the workflows tab, we have this categories, which kind of helps you, sort down the list if you know what you're looking for. If you don't, you feel free to browse all. There's also an ability to search for specific workflows. For example, RNA-seq, and it will pull up anything related to RNA sequencing. Now, when I'm ready to launch something, I will click on whatever workflow it is that I want to launch, and it will pull up a page similar to this. Every one of our workflows is built to give you an ease of use, so a lot of them follow similar page setups. Most of them will have an overview page discussing a summary of the workflow itself, the different steps, and some information and metrics on how to use the workflow and why we made the decisions we did. There's also going to be input and output tabs, as well as citations marking any tools or references we use to generate these workflows. In the top right corner, you will see the version option. We keep all of our versions and maintain versioning as we go so that there is always reproducibility for any experiment you might have done at any point in time. Once you select the version, you can go ahead and hit Run Workflow, and that will launch the workflow you selected. In this case, we're doing some RNA-seq. So here, there is essentially a layout of multiple pages, each with different inputs that you must provide to the workflow. The idea here is that we made it quick and easy, point and click, to do high-level analysis on your workflow. So for RNA-seq specifically, I can pre-select some different input options, as well as select the input data that I wish to run this on. I can also select a sample sheet or with metadata to accompany the input files that I'm going to be running an analysis on. Once I do all the steps per page.
I have a question. What if I don't have a metadata file? How do I know what the right format is?
If you don't have a metadata file like we have provided here, you can always create a new one. And you can do that here. There's always going to be a create new file, and we display the needed metadata information that you must provide. Much like an Excel sheet, you can copy and paste, you can add rows, and you can remove rows. And this will be a layout for a design file you need to create, a metadata file you need to create on the fly. However, in this case, we have already pre-selected one.
And can I just ask if I had in the metadata file, and you can just show in this example, how would I add an extra sample to those
Sure.
to my existing file?
If I had a file that already existed like this, and I wanted to add a new sample, I could simply insert row and make the necessary sample changes that I wanted to include. And I would have to, of course, fill in all the necessary metadata. And once that's done, I'd simply hit the save button. The save button makes sure that the changes that I've made are now saved to the design or the metadata file. And it would be as simple as that. In this case, I'm going to remove this since we do not actually have samples matching. Once you've completed all the steps on any given page, you can go to the next step. As you can tell with RNA sequencing, you can select the genomes available. We try and host as many genomes as possible, as well as different annotation versions for each genome. Once the genome is selected, we can go to some advanced config options. For a typical user, we try and set the most common and good practice defaults for all of our advanced configurations. However, for those that want some more freedom with the choices in the analysis, they are more than welcome to change any and all of the config, the advanced parameters. So that includes the ability to, trim or not trim, different variant calling, and other inputs that are relative to specific tools used in the workflow. Once all the advanced configs is set, you can give it a name on the last page, and you can check any of your inputs to make sure these are the ones you wanted. If you need to go back and change anything, you can always do that and go back, make sure the changes are what you want. And finally, in the end, you can hit run workflow. Now, with the workflows, we've made this activities page, which is what you see here. Any workflows that have been run or are running will show up on this activities page. This page is kind of a quick summary. It's searchable, so you can search by user, you can search by time, name, status. You can also sort and filter these however you would like. Each of these runs will allow us to keep track of the different runs that we're have going on in the project. In this case, this is the run that we just launched. You can see that we have these different tabs for quick viewing. Status is a quick update of where the workflow is. At this point, it's still running and getting ready, so none of the steps have finished. And you can see that there is a log section as well that we can keep track of. I'm now going to show you a finished version. Here you can see this is a completed RNA-seq run of the same data. First, we have a results tab. This hosts any HTML result pages that we want to quick display to the user. As you can see here, we've got a lot of different QC and analysis information. In this case, around RNA-seq, we have FPKMs and count tables, heat maps, all the necessary QC information. We also provide with many and most of our workflows a multi-QC report, giving the user more, information and confidence in their sequencing and the reads that they have just analyzed. Obviously, we do have many other output files that are available to the user, including your typical count tables and different R package outputs that can be imported and used by the user. We also include tons of QC outputs from all of our workflows, and along with the results tab, we have the status tab. This shows all the completed and errored processes that occurred in the workflow, so you can keep track of what step the workflow's on. Files has all the output files. Here it is a quick link to your data folder, so you can easily copy locations, move them around, and open them in the data tab of the platform. We also have a quick input summary for the user so that you can go back and look at your previous runs. And let's say you get a new batch of sequencing or a new set of data you want to run the same way, you can always go back and reference how you ran it and what your different parameters that you selected were for each individual run. Finally, we have our logs tab. This is just for the user to kind of trace logs and be able to, debug and check different issues. It's also for our form bio team to be able to assist you in any and all errors that you may occur. Now, with the results tab, some of our workflows have the advanced capability of a analysis, an interactive analysis. Those would have an open analysis button in the right upon the completion of the workflow. Just like this. Once you click it, it will launch it. Sometimes it takes a bit of time for the interactive visualization to spin up, but once it does, you will get a green "Your analysis is ready" check mark, and you can go ahead and click the tab. That will launch one of our interactive visualization tools. In this case, this tool is an RNA-seq tool. Each of our tools is custom built to that specific workflow, and thus some workflows have this and some do not. In this case, this is a quick walkthrough of all the different data that was created and analyzed through our RNA-seq pipeline, and would allow you to walk through your data step by step and really break down and understand, give you a deep understanding into the data that you ran analysis on. This is great to draw scientific conclusions on your data, as well as give you insight into your samples themselves. As you can see, it gives you the ability to compare samples. We do differential expression analysis, as well as gene set enrichment analysis, and many heat maps. All of these are based on the necessary metadata that you provided, but all of our data is here for your disposal, as well as in the file form that you have seen on the platform itself.
Jeremy, can I ask you a question about the open analysis?
Okay.
If we are there? There's necessary metadata, but if I have extra metadata and I might want to compare my samples against that extra metadata, can I do that in that space?
Yes, if assuming you provided all the extra metadata, you can compare any of the extra information that you provided. Yes.
Okay. For example, if I had extra fields and I wanted to compare the abundance of a gene across those extra fields, I could do it by that extra metadata field.
Yes, that is correct. And if you provided, for example, let's say in your metadata, along with the group and ID, you provided a treatment or a cell type, you could then here in the analysis be able to compare by cell type as well as group and ID. And this goes for any and all metadata columns you wish to provide for all of your samples. So going back to our workflow page, all of our workflows are built to function similarly to the workflow I just showed you, such that if you were to go to any workflow, for example, if we wanted to call variants on our whole genome sequencing, we would be able to do so, and it would be a familiar layout. This way, it is easy for any user to quickly use and learn how to use any of the tools and the workflows that we provide on the platform. All of them inputs will be functionable the same way, and they will all have some help text to go along with them, whether they be in the overview docs or on the right side of the workflow launched tabs. There will be help text to go along with each of them to walk you through the different steps, but to keep familiarity with being able to use the tools. A lot Sometimes with our biology and our science, we don't need a heavy analysis run on samples. Sometimes we need simple information. And for that, we at Form have developed our toolbox tools. Similar to the workflows, the toolbox tools are quick, easy, one off steps that are just, you could think of as a multi tool for a biologist to quickly get things done while they're trying to answer the questions that their samples hope to provide. In the toolbox, you can see we have a bunch of tools available for the user, all doing different things, such as simply aligning two sequences or calculating sequence length. Similar to our workflows, all of these are set up to look and function similarly. So, for example, with sequence length calculation, you can either give a FASTA input, or you can simply drop a sequence in here, and it will calculate these lengths for you on the fly. Obviously, this is small, but it would work for very large sequences as well. All of these run extremely quickly and give you back results within minutes. Some of these are tailored tools, some of them are more general. Sequence length, calculate percent GC. Some of these are more advanced tools like G-block synthesis and engineered peg design for prime editing. We also have the ability to just quickly design primers for your PCR or convert a sequence from DNA to protein. The toolbox is essentially here to be that quick, everyday helper for the scientists to get back to their analysis and focus on the results. With the toolbox, we also have our form bench. The form bench is for the more sophisticated bioinformatician user. This has access to some more advanced tools that aren't necessarily point and click, but provide a lot of freedom and what do they do? Provide for a more custom analysis for the scientist. So some of these we have discussed before, such as data transferring and the form shell. But some of these are a little more interactive, such as our visualizations. We host CellXGene from Chan Zuckerberg, which is a single cell tool for visualization. Sanger Analyze R allows us to analyze and view Sanger data, and our TensorFlow is our quick visualization toolkit for you. We also have access to Jupyter Lab and our Studio IDE. This allows the scientist or user to be able to write some of their own Python and R code while still remaining within the confines of Form and the analysis that they've done here, making it easy to further investigate the data and samples that the scientist has analyzed on the platform. Finally, a lot of this can be covered in our documentation. That quick links at the bottom and helps answer any questions that you may have regarding our platform. We look forward to helping you get the most out of your data and answer as many scientific questions as you can.